Welcome back everyone. I hope you have had some great interactions with my colleagues in exhibit booth. Now I would like to invite our next speaker, Dr. Tomoji Mashimo from Institute of Medical Sciences, University of Tokyo, Japan. He is the director of Laboratory Animal Research Center. Topic of his talk is rapid and accurate detection of novel coronavirus SARS-CoV-2 using CRISPR-Cas3. Over to you, Dr. Tomoji Mashimo.
First of all, I would like to thank the organizer for giving me this opportunity, especially in this critical condition of COVID-19 here in Japan. This year, April, actually I have moved to the University of Tokyo Institute of Medical Science from Osaka University, then faced this difficult situation. So today, I would like to talk about CRISPR tools for genome editing as well as for new diagnostic tool for COVID-19 that we have recently developed using CRISPR-Cas3 systems. In this first slide, I would like to confirm my COI. I am a co-founder and now scientific advisor for C4U startup company here in Japan. First episode in my talk is CRISPR-Cas3 system is useful for genome editing in eukaryotic cells. As you know, CRISPR-Cas9 is widely used as a gene editing tool all over the world, but it is just one in a wide variety of many CRISPR-Cas systems. For example, there are several Cas proteins from Cas1 to Cas14, which are classified into two classes, Class 1 and Class 2. They are further divided into three different types from Type 1, Type 3, Type 4 in Class 1, and Type 2, Type 5, Type 6 in Class 2. What are the difference between the two classes? The main difference is the number of the effector molecules. Class 1 effectors contain multiple subunits as you see here. Class 2 effector are single large proteins like Cas9, Cas12, and Cas13. Cas9, as you know, belong to Class 2 Type 2 system, and Cas12, also we call CPF1, is Type 5 system for DNA editing. Cas13 Type 6 system enables the RNA editing, which are different from the Cas9 and Cas12 DNA editing. So far, Class 2 system Cas9, Cas12, and Cas13 have been used for genome editing. Then we had a question, can we use Class 1 CRISPR system as a genome editing tool? The answer is yes, we can. Class 1 Type 1 CRISPR system we call CRISPR-Cas3 system contains 6 Cas proteins, Cas5, Cas6, Cas7, Cas8, Cas11. Those 5 Cas protein complex called cascade. Cas3 has a strong DNA nuclease and helicase activity that could degrade phage plasmid DNAs. Cascade contains guide RNA or called CRISPR RNA shown in yellow line that recognize target sequences. The different features between Cas9 and Cas3 system for genome editing are several. As you can see here, the CRISPR-Cas3 induces large deletions upstream of the target sequences. The deletion size ranging from several hundred to thousand base pair, you can see degraded PCR product, PCR band on agarose gel here. And you can also see several hundred to thousand deletions bars here upstream of the target site as shown in red line. Therefore, the Cas3 is more applicable to delete large lesions or knock out full genomic region. On the other hand, Cas9 generally used for generating small indel mutation. Another feature of Cas3 is CRISPR RNA recognition size, which is totally 27 nucleotide, longer than 20 nucleotide of Cas9 guide RNA. So we hypothesize this longer recognition of Cas3 CRISPR RNA may induce more specificity of genome editing. In this slide, we compare the two systems Cas3 and Cas9 in the same experimental conditions for evaluating the off-target effects. We identified both Cas3 and Cas9 mediated on-target mutations, as you can see in red dot. However, we only identified off-target mutations induced by Cas9 here in blue dot, but not off-target mutations observed by Cas3 genome editing. We can also find various small indel mutations by CRISPR-Cas9 here, you can see. So, type 1e CRISPR showed comparable or lower off-target activity compared with CRISPR-Cas9. In this slide, we have tried a functional gene knock-in using CRISPR-Cas3 systems. We used donor plasmid vectors including EF1 alpha promoters and purogene cassette with 1 kilobase homology arms at both ends. And we transferred those donor plasmid into the HEC293 human cells with the CRISPR-Cas3 systems. As you see, both CRISPR-Cas3 and Cas9 system can induce efficient gene knock-in at the target site by homologous recombinations. Finally, collaborating with Hotta Sensei and Okuzaki Sensei at IPS Center in Kyoto University, we showed exon skipping of the Duchenne muscular dystrophy gene using CRISPR-Cas3 system as an example of potential therapeutic applications. We compared the efficiency of Cas9-mediated exon skipping using two guide RNA, as here blue bars, or Cas3-mediated exon skipping as shown in red bars here. We found that even a single CRISPR RNA with Cas3 drastically increased the exon skipping efficiency, as shown here red bars, compared to the dual guide RNA Cas9 system, as shown here blue bars. Because Cas3 could mediate large deletions of the genomic region including the fold exons here. We also confirmed the recovery of DMD gene expression in the patient-derived iPS cells. As you can see here, messenger RNA levels by RT-PCR and protein levels in Western blotting gels, the CRISPR-Cas3 recover DMD expression in patient-derived iPS cells. Here is a summary, a character list for Cas3 and Cas9 mediated genome editing. Cas3 has a strong helicase and nuclease activity which can easily induce a broad DNA degradation, like unidirectional large deletion at the target site. Therefore, it may be used for complete functional gene knockout or for excluding the large genomic regions such as promoter regions, enhancer regions, and repeat sequences. The importance for the industrial companies is the patent issues, I think. It might be more clear for CRISPR-Cas3 compared to the Cas9. Here in episode 2, I would like to talk about CRISPR-based diagnostic tools Cas12, Cas13, and Cas3. As you know, Jennifer's group and Fan's group have harnessed Cas12 and Cas13 respectively to make CRISPR-based diagnostic tools. They have started the venture company Mammoth Bioscience and SHERLOCK Bioscience and successfully developed rapid nucleic acid detection system called DETECTR with Cas12A and SHERLOCK with Cas13. They actually used unique feature of the CRISPR-Cas enzyme. This is both Cas12 and Cas13 enzyme, but not Cas9 enzyme exhibit non-specific end nuclease activity in trans after genome editing at the target site in cis. Using this collateral single-strand DNA cleavage activity, they developed easy and rapid detection system of virus infections such as COVID-19. So my question is, how about Cas3 systems? Here you can see the CRISPR-Cas3 system has also collateral ssDNA cleavage activities. When we mix cascade Cas3 a double-strand DNA, that is the target double-stranded DNA fragment, and donor DNA, that is either linear single-strand DNA more than 1 kilobase, or circular M13 single-stranded DNA. We observed the single-strand DNA cleavage for both DNA. If the target sequence perfectly matched with the target double-strand DNA and Cas3 was activated. You see here DNA degradation on the agar shell by red bars. This is a representative protocol for SARS-CoV-2 detection, including 4 steps. First step is a conventional RNA extraction. Second step is RT-LAMP for DNA amplification step. This just needs 20 to 30 minutes. Third step is Cas3-based nucleic acid detection in 10 minutes. And lastly, lateral flow assay in 2 minutes. Totally, 40 minutes we can detect SARS-CoV-2 virus with excellent sensitivity and high specificity as compared to RT-PCR-based common COVID-19 assay. Therefore, we named this protocol Cas3-operated nucleic acid detection assay as CONAN. CONAN used a lateral flow assay test strip, which is generally used for portable antigen system like influenza virus detection. As you can see here, CONAN can detect rapidly SARS-CoV-2 virus. Without infection of SARS-CoV-2, you would see lower negative band here. If you infect it, you would see a positive band in the upper side here. For those data, we have already reported a preprint at NetArchive. You can read it on the website. And released from the homepage of the University of Tokyo, here you can see. If you are interested in our technology using CRISPR-Cas3 systems for genome editing and CRISPR-based diagnostics, please contact the startup company C4U. Here is the website. So thank you very much for your attention, and I am very glad to have your questions.
Thank you, Dr. Mashimo, for sharing exciting approach of using CRISPR-Cas3 for COVID-19 detection.